Oncolytic viruses work by priming the immune system in a manner similar to a vaccine. The treatment involves the injection of oncolytic viruses into an accessible tumor site, primary or metastases. The viruses would then infect tumor cells. They would deliver their DNA into the cell, which then takes over the cellular reproductive mechanism to produce tens of thousands of copies of itself. The virus presents a strong danger signal to the immune system through the release of cytokines and chemokines. Oncolytic viruses would then cause lysis of the tumor cells, releasing tens of thousands of copies of the virus that then infect other tumor cells. When the virus replicates in tumor cells and lyses them, it releases hidden tumor-associated antigens. These new, previously hidden antigens are now recognized by antigen-presenting cells, most commonly dendritic cells. The dendritic cell's main function is to process antigen material and present it on the cell surface to prime naive cytotoxic T cells against these tumor-associated antigens. These newly primed T cells would then go on to attack and kill other tumor cells that have the same tumor-associated antigens not previously recognized by the immune system. PsiVac is a conditionally replicating adenovirus serotype 5. It infects tumor cells and lyses them extremely effectively. At the same time, it's unable to lyse normal cells and cannot replicate in them. How does it achieve this? Well, let me first tell you that adenovirus 5 is the common cold virus. It's the most studied virus and used to study the host immune response. The adenovirus has evolved over millions of years. Its evolution started with the early genes, which are extremely important regulatory genes, whereas the late genes are important for structural integrity of the virus. To achieve selectivity, others have deleted parts or all of the early genes. This, we now understand, weakens the replication efficiency of the virus. PsiVac has no deletions and is nearly identical to the wild-type virus. It was designed with a mutation that maintains its structural integrity and hence its efficacy, as well as maintaining our goal of inhibiting replication in normal cells. How is this achieved? Our unique approach is to introduce a single base pair mutation at a specific splice site in the E1b region. This single base pair mutation, we have been able to demonstrate, enhances selectivity without hampering the replication efficiency of PsiVac in cancer cells. So how does PsiVac selectivity compare with other viruses? Here we see replication assay in normal human epithelial cells. PsiVac replicates slowly and is significantly more effective than Onyx 015, and certainly better than the wild type. We therefore see that it is certainly safer than the commercially available Onyx 015 H101. We know it's safe, but what about its replication in tumor cells? Here we compare PsiVac replication in non-small cell and large cell lung carcinoma. We can see that PsiVac replication rate is as good as the wild-type control and significantly better than Onyx 015 H101. We then decided to test PsiVac in 16 solid tumor cell lines to assess how well it replicates. We were delighted to see that the virus replicated extremely well in every other solid tumor: pancreatic, gastric, melanoma, colorectal, glioma, ovarian, and fibrosarcomas. We then conducted in vivo studies on immune-deficient nude mice with human non-small cell lung carcinoma injected into the flank. We see that the control of tumor growth is as impressive with PsiVac as the wild type and superior to Onyx 015 H101. Median time of survival was determined by Kaplan-Meier survival analysis. 2 mice out of 7 were cured with PsiVac, which is comparable to the group treated with the benchmark, which is a wild-type virus. PsiVac is safe, is powerful, and is replication efficient in all solid tumor cell lines tested so far. Heads of terms have been signed for collaboration with Lonza to share costs of production. A signed LOI agreement to start pre-development work, which includes material and documentation transfer, as well as assay testing, will pave the way for cGMP production thereafter. Planning is underway for a Phase 1-2 clinical trial to be conducted at the Royal Marsden and University College hospitals. Professor Kevin Harrington, a world expert in oncolytic virotherapy, is the lead investigator for these clinical trials.